Chapter 5 Microbial Metabolism. So let's start today's lecture with a case study. Brad is an active 15-year-old high school student who scraped his knee skateboarding. He washed his knee well with soap and water when he got home and didn't think any more about it. The next morning, Brad's knee is stiff, is swollen, is quite red. He washes it again when he takes a shower. Later on, he develops a fever and his mother takes him to the doctor. The doctor examines Brad's knee and tells him that the wound is infected. The doctor is concerned by the rapid progression of the infection and Brad's fever. She admits Brad to the hospital with cellulitis and orders blood cultures followed by an intravenous course of antibiotics. After 2 days of treatment, his knee actually looks worse. The infection is spreading to the surrounding tissues. So, what's at work here with Brad's infection? Well, pathogens can infect us in various ways. Once we're infected, we either clear the infection or get sick or suffer symptoms. Pathogens must obtain nutrients and produce energy to cause the adverse outcomes that we experience. Let's take a closer look at the pathogenic process. All organisms and microorganisms are able to carry out their functions based upon a chemical process called metabolism. Well, what is metabolism? Metabolism is the sum of all the chemical reactions that take place in an organism. That includes microorganisms as well, bacteria, etc. Metabolism actually includes two processes, catabolism and anabolism. Catabolism is a process that breaks down complex molecules into simpler molecules, stores energy from these simple molecules in the form of ATP. This energy is later used to power anabolism, and it also makes precursor metabolites for anabolism. Anabolism is a process that builds complex molecules from simple molecules. So, generally speaking, when we think of metabolism, we think of metabolizing our food. And to a degree, that is correct. That's a correct assumption to make. But it goes a little deeper than that. In the process of metabolism, that combination of catabolism, breaking down large macromolecules to smaller ones, and anabolism, taking smaller ones to be put together to build bigger ones. What we do is when we consume food or whatever source nutrition comes from, and when I say nutrition comes from, be it liquid nutrition or the nutrition that various microorganisms may need to obtain, that chemical energy that we obtain from these nutrients like carbohydrates, fats, proteins, and other macromolecules is then used to produce energy. That energy is in the form of a molecule called ATP, which stands for adenosine triphosphate, and we'll talk about that in more detail a little bit later. And then once we metabolize this food via these various chemical processes and chemical pathways, we will eliminate waste, and generally that waste may be in the form of carbon dioxide or water. We also generate heat in that process of metabolism. So I already stated that catabolism is breaking down these large molecules into smaller ones. And when this happens, energy is released. So the reactions that are involved in these catabolic processes are called exergonic because they release energy. Whereas anabolism, where we are synthesizing or building larger molecules from individual components, those reactions actually require energy. So they are said to be endergonic. So where is this energy coming from that I'm talking about? Well, the energy is actually coming from electrons that are located within the atoms of the molecules of these different macromolecules like proteins, etc., or carbohydrates. And so what happens is energy, as I've stated, is stored in the electrons. The electrons are then transferred between molecules of different atoms. There are two processes that go on here. One is called oxidation, and the other one is called reduction. So we refer to this overall process as reduction, oxidation, or redox reactions. In the process of oxidation, there is a loss of electrons. When electrons are lost, that means that atom becomes less negative and energy is actually released. But in the process of reduction, electrons are gained, and this requires energy. So what we have at play here is we have what's called the electron donor molecule. The electron donor molecule is the molecule that has an overall negative charge and it's giving up an electron. When it gives up that electron, it becomes less negative. So we now say that that molecule, after it has given up the electron, is oxidized. But the electron acceptor, once it receives, it may be oxidized, but once it receives the electron, now it has an overall negative charge, and we now say that it is reduced because it is more negative. So understand that oxidation reduction reactions or redox reactions actually are carried on simultaneously. One does not happen without the other. There has to be an electron donor and there has to be an electron receiver or acceptor. So electrons can carry energy. Energy transformation occurs in redox reactions with transfer energized electrons from one reactant to another. And again, this occurs simultaneously. This whole redox process of oxidation and reduction, transferring electrons or donating electrons and accepting electrons. So to drive the point home of the whole redox process, let's just kind of use a different example. Let's look at it in the form of, say, gift giving. If you give someone a gift, and in this case, the electron donor will be the gift giver. So that gift giver is now losing the gift or giving up the gift or donating it to someone else. Whereas now what ends up happening is when the donor gives up the gift, which in this case is the electron, you have a loss of an electron, therefore a loss of energy from the molecule, atom, or ion. The electron donor now becomes oxidized because it is less negative. So the gift giver is oxidized. She's energized. She's elated that she's given the gift to someone else. And the electrons are now transferred from donor to acceptor. Now, in the case of the electron acceptor, that's the person receiving the gift. That is that person receiving that energy. So there is a gain of an electron. And this gain of energy from the atom or molecule now is accepted by this electron acceptor or gift acceptor. The electron acceptor now becomes reduced because it is more negative. So this gift receiver, this acceptor, this electron acceptor humbly receives the gift. So kind of, bowed down a little bit. So in other words, reduced, humbled. That's how I like to kind of look at it. I thought that was a neat way, a neat spin to put on it. So remember that these redox reactions occur simultaneously, and there's an acronym that you can use to kind of help you remember with redox which one is giving and which one is accepting. And the acronym is OIL. Oxidation involves loss, OIL. Oxidation involves loss. Loss of what? Loss of an electron, which means loss of energy. Reduction involves gain, RIG. Reduction involves gain. Reduction in that it becomes more negative after taking on these electrons, thereby making this acceptor molecule more negative. So redox reactions happen simultaneously with an electron donor and an electron acceptor. So just kind of remember that acronym OIL to kind of help you remember the flow of electrons from donor to acceptor. Now, again, when we spoke of atoms, we talk about atoms having subatomic particles, those subatomic particles being protons, which are positively charged, electrons, which are negatively charged, and neutrons, which are neutral. They have no charge. Okay, the protons and the neutrons are located within the nucleus of the atom, but the electrons actually orbit. In other words, kind of rotate around or orbit, occupy a space around the nucleus of the atom. So electrons don't usually exist just free in cytoplasm. They orbit the nucleus of an atom, and cells use electron carrier molecules to transport electrons from one place in a cell to another. So they don't just jump from one place to another on their own. they need some assistance, and the assistance is in the form of molecules that are called electron carriers. So there are three important electron carriers that we find in various biochemical processes. We won't get deep into the woods. I just want you to have a surface level knowledge of this. But you have what's called nicotinamide adenine dinucleotide, or NAD+, nicotinamide adenine dinucleotide phosphate, so it means it has a phosphate group on it, flavin adenine dinucleotide, FAD. These are derived from vitamins. Cells use these molecules in specific metabolic pathways to carry pairs of electrons. So I already stated that these redox reactions occur simultaneously. I also told you when we were talking about electron donors and electron acceptors that these electrons don't just jump around on their own, but instead they have what's called electron carriers that help them to move from place to place. These processes are always going on continually within our system so that our cells are continually functioning. The electron transport chain, there is a system within our cell membranes that's called the electron transport chain. This is a series of sequentially linked oxidation reduction reactions. Energy released at each step cells store for use in chemical reactions. Photosynthesis and cellular respiration are going to be two processes that we talk about that we can see how this whole redox reaction process works. So I think we all have a loose understanding that nutrients contain energy. Well, this energy is stored in the chemical bonds of these nutrients because remember, be it proteins, carbohydrates, etc., they are made up of molecules. Those molecules are made up of atoms, and these atoms bind together to make up the molecule. So energy is stored within the chemical bonds of the nutrients or of the molecules of the nutrients. Cells cannot use the nutrient energy in its stored form. Metabolism breaks chemical bonds, causing this energy to be released. It's an exergonic reaction. And this released energy is used in anabolic pathways or anabolism, which is an endergonic, requires energy, an endergonic reaction to synthesize or to make new compounds. So again, just kind of keep this picture in mind. You already saw this slide that when we talk about metabolism, we are talking about various processes in which chemical energy is drawn from other molecules like carbohydrates, fats, proteins, etc., in order to draw energy from the bonds that are actually located within these macromolecules so that we can now produce the energy that we need, or our individual cells can produce the energy that it needs in the form of ATP. So this cellular energy that we talked about that is in these bonds of the micromolecule is in the form of ATP. So ATP stores energy in a form that cells can use. ATP stands for adenosine triphosphate. And the tri, anytime you see the prefix tri, three, so three phosphate molecules. And they are held together by chemical bonds. ATP also has a sugar called ribose, as well as a nucleotide adenine. So just to kind of give you a pictorial representation of what's going on here, ATP is coupled with chemical reactions. So these redox reactions that we talk about. So reactions that break down ATP are coupled with reactions that require energy input. When reactions are coupled together, energy released from the ATP can power the second reaction. So we'll consume the food, which has various macromolecules in it, like carbohydrates, proteins, etc., in it. And then once we consume it, these bonds are broken within these molecules. And when those bonds are broken, it will release actually ATP as well as energy. And that energy now can be used to do other things. In this particular example, this person is riding a bike. as a result of the energy he has obtained from consuming these nutrients that he's gotten energy from their broken bonds. So these various reactions don't just spontaneously happen, they need a little help also. And they receive help by compounds called enzymes. Now, most enzymes are proteins, not all of them, but enzymes are a specialized type of protein, but we'll see that there are other types of enzymes as well, and they serve to speed up chemical reactions. They're not consumed during a reaction. In other words, they're not used up. If you start off, if there's such a thing as having 5 units of enzymes, when the reaction starts, there's 5 units of enzymes. When the reaction ends, there's still 5 units of enzymes because these enzymes are not consumed or changed during the process of these chemical reactions that they speed up. Whenever we talk about something speeding something up, it is a catalyst. So enzymes are a catalyst. A catalyst is something that will start the reaction and cause it to go forward. So, and how the enzymes work, and we'll see a slide better depicting this, is they bring these reactants, so the different things that are working together in a reaction, they bring them together or closer together, and this causes a lowering of their activation energy. In other words, not as much energy is needed for this reaction to go forward. So the macromolecules in this particular case, let's just say a carbohydrate or a protein, we're going to give it a generic name and call it a substrate. And we've already talked about these macromolecules, be it a carbohydrate or a protein being bonded together, that their atoms are bonded together to make these molecules, right? So now these macromolecules, which we will refer to as substrates, they need to be able to bind to the enzyme in order for the enzyme to affect it. It has to bind to a specific place on the enzyme. That specific place where that substrate binds is called the active site. Once a substrate is bound to the active site on the enzyme, now the processes go forward that allow these bonds to break. So combining the substrates with the enzyme, you form what's called an enzyme substrate complex. Once that complex has been formed, it activates the enzyme to now go forward with its processes. That enzyme will cause chemical reactions to move forward that will break the bonds of those macromolecules, and now you will end up with 2 different individual products or smaller units. So each enzyme fits the shape of a particular substrate. So an enzyme that works on carbohydrates will not work on breaking down fats and vice versa. They are very specific. So the substrate is what the enzyme acts on. So be it carbohydrate, protein, etc. Substrates are able to bind to the enzyme's active site where the chemical reaction occurs. So again, the place on the enzyme where the substrate will bind is called the active site of the enzyme. So I mentioned earlier that enzymes lower the activation energy. So each chemical or biochemical reaction has a certain amount of energy that it needs in order for it to go forward. What an enzyme will do is it will actually bring those reactants together, kind of like that picture we saw where they fit in snugly, and so now they're kind of sitting close together in the previous slides, the components of the substrate. This enzyme now, once the substrate fits into the enzyme's active site, it actually brings these substrate components close together. So now you've got closer proximity for the reaction to occur, so not as much energy is required. Hence, the enzyme lowers the activation energy by bringing substrates closer together so that the reaction moves forward more quickly. So, not all enzymes work in the same way. There's various enzymatic activity. One is called negative feedback or feedback inhibition. Negative feedback or feedback inhibition is a change that triggers an action that reverses the change. So, in other words, something happens, and whatever that something is that happens, it can trigger another action to cause that first thing to not continue. So, in other words, if you're hungry, your stomach growls, you eat. When you get full, now you stop eating. Okay. So that negative trigger, that negative feedback is okay, when I get full, stop eating because that has signaled my brain that my glucose levels have now reached homeostasis. They are back in balance. Now, enzymatic activity can also be what's called non competitive or competitive. When we speak of non-competitive and competitive, what are we competing for? We're competing for that active site that's located on the enzyme where the substrate will bind. So in the case of non-competitive inhibition, whatever the inhibitor is, or something that's kind of competing with a substrate for the site, it doesn't compete with the substrate because it does not bind in the same site that the substrate would normally bind. It doesn't bind in the active site where the substrate will bind. Instead, it will bind somewhere else. When it binds somewhere else, it changes the shape of the active site. Now, the substrate cannot bind because it was looking for a partial square to sit in. But now that inhibitor, that non-competitive inhibitor, has bound somewhere else, causing the shape of the active site to change. And so now the substrate won't fit there. So it's not competitive in that it's not binding directly to the active site, but it is preventing the substrate from binding to its normal active site on the enzyme. In the case of competitive inhibition, whatever this inhibitor is, and it could be like a drug or a therapeutic drug, like, I don't know, aspirin or Tylenol or whatever. It could be anything. It could be a microorganism like a bacteria. Whatever that competitive component is that's responsible for this competitive inhibition, it is competing with the normal substrate for the active site. So it will actually, it may not be an exact match, but it's close enough to the shape or resembling what the normal substrate would look like that it will bind to that active site. And once it binds to the active site, guess what? The regular substrate or the normal substrate can no longer bind. So enzymes are very sensitive to their environment. They can be degraded or changed by various environmental conditions, the temperature. If you have an enzyme that works at a particular temperature, let's say within our bodies at 37 degrees. Well, if that environmental temperature gets too hot or too cold and it's not optimum for the enzyme, then it won't work properly. pH concentration, which is just the concentration or amount of hydrogen atoms within a cellular environment, if the pH isn't just right. We say that pH 7 is neutral. The optimum pH for enzyme activity is 7.2, so right around neutral. Neutral. If the salt content, if it's too much salt, if it's a very salinic environment, that could impact enzyme activity. So can drugs. Understand this, enzymes, especially those that are proteins, their activity level also is based upon how they are folded. So just like with proteins, the folding of proteins, uh, tertiary, quaternary, or what have you, that determines what type of protein it is and how it will act and what it's used for. Similarly, enzymes are the same way. The folding of those proteins or those enzymes. And when that structure of folding of enzymes are interrupted or broken down, that process is called denaturation. So denaturation is a deactivation of enzymes based upon its structure being interrupted. And we see that this structure can be disrupted by various things, temperature, pH, salt, drugs. As a general rule of thumb, when we look at the names of enzymes, Enzymes generally have the suffix ase on the end, but they also tend to give you a hint in terms of either how they work or what they are working on because it is incorporated into the name. So, hydrolases, they catabolize molecules by adding water and breaking these molecules down in a process called hydrolysis. Whenever we see the word hydro, we think of water. When we see the suffix lysis, Lysis, we think of a splitting or a breaking down. Lipase, it ends in ASE like other enzymes, and it breaks down lipids. Or, for instance, uh, oxidoreductases. They're involved in that redox production, and they can remove electrons, therefore causing oxidation, or they can add electrons, causing reduction of a particular atom. So again, there are hints within the name of an enzyme as to either what it works on or how it works. So I mentioned a couple of slides back that some enzymes are RNA enzymes, and those are called ribozymes. So enzymes that are composed of RNA are called ribozymes. Most enzymes are proteins and they function independently. But there are some enzymes that need help to become active. They aren't active and aren't capable of doing their job unless they have something else attached to it. So we have what's called apoenzymes. These types of enzymes are composed of protein and non-protein portions. Cofactor is a non-protein component of these apoenzymes, and it can be almost anything an inorganic molecule, which means it doesn't contain carbon, like iron, magnesium, zinc, or it can be organic, which means it does contain carbon, a carbon atom, which is usually bound to hydrogen. If it is organic, then we say that it is a coenzyme. So if the cofactor is organic, then it is called a coenzyme. And all coenzymes are either vitamins or they contain vitamins. So all coenzymes, which are just organic cofactors of apoenzymes, either are themselves vitamins or they contain vitamins. So these apoenzymes are inactive. They are an inactive unit until a cofactor binds to it. And again, the cofactor can be inorganic, like iron, magnesium, etc., doesn't contain any carbon, or it can be organic, okay, and if which contains carbon, carbon bound with hydrogen. If it's organic, then it is called a coenzyme. And remember that coenzymes are either vitamins or they contain vitamins. Now, when you have everything put together, say for instance in this apoenzyme, it's called a holoenzyme. So kind of like holoenzyme is composed of the apoenzyme as well as its cofactors. So a holoenzyme is an enzyme that is complete. It is an apoenzyme or a complete apoenzyme. It's an apoenzyme which also has its cofactors attached and therefore now it's active and ready to go. So a couple of slides back, I told you that NAD plus, NADP, and FAD are electron carrier molecules that we see very active in these redox reactions. Well, guess what? They also can function as coenzymes as well. So just a reminder of a previous statement that I made and kind of a different look. Enzyme substrate fit is very specific. And we speak of enzyme substrate specificity as well as induced fit. You know, there is a concept that's called lock and key. It's not used as commonly now, that term is not used as commonly, but basically the idea is that the key that unlocks the door at my house shouldn't unlock the door that's at your house. So enzymes and substrates are very specific in that they only work on one specific thing. So one particular enzyme works on one particular substrate. Enzymes are very specific. The enzyme active site changes slightly when it binds to its substrate. And that's where that whole concept of induced fit comes along because, yeah, it may have the general shape, but it may not be the perfect size. But when that substrate binds to the active site, then if it needs to, it kind of forces its way, and now the active site will just basically conform to that substrate, and now it's what's called an induced fit. So that fit is kind of stimulated, if you will, by the actual binding of the substrate. When the enzyme and substrate bind, when that substrate binds to the active site of the enzyme, we say that an enzyme substrate complex is formed. Bonds within the substrate are broken via catabolism, so broken down. Metabolism is a metabolic process that breaks down large molecules to form two new products. Enzymes dissociate from the newly formed products at the end of this whole process. And once those new products detach from the enzyme, because now all processes are finished, the enzyme will resume its normal shape and normal configuration and wait for the next substrate to come and bind. So on one of the previous slides, when I talked about competitive and non-competitive inhibition, and I said that in competitive inhibition, the substrate will bind to, or the competitive substrate will bind to the active site of the enzyme of the normal substrate. But in non-competitive, you have cases where the competitive substrate will bind to a different site and cause the active site of the enzyme to change shapes. This particular scenario, when you have a competitive substrate binding somewhere other than the actual active site of the enzyme, but binding somewhere else on the enzyme and causing that active site shape to change, that is called an allosteric reaction. So now that new site is called an allosteric site. That site where it binds to cause the active site to change is called an allosteric site. So an enzyme can be activated by the binding of a cofactor to a site other than the active site. Allosteric reaction causes the active site to change shape. It's non-competitive because it's not competing directly with the active site of the enzyme. It's binding somewhere else, but it does cause that active site to change shapes. And this could be anything. The binding of a heavy metal ion or some cofactor can cause this allosteric activation. And the allosteric activation, of course, is, changing of that active site. But also, there are some microorganisms that can bind allosterically. Say, for instance, E. coli or Escherichia coli. It can actually bind allosterically to prevent certain processes going forward within our cells. So we now know that the word catabolism is just one of the processes involved in metabolism, and the process of catabolism specifically breaks down larger macromolecules like carbohydrates, lipids, proteins, etc. So in carbohydrate catabolism, keep this thought in mind, many organisms use carbohydrates as their main form of energy. Primary carbohydrate used for energy is glucose. The primary carbohydrate used for energy is glucose. There are other sugars as well involved, as well as other molecules and compounds like amino acids and fats. These things are also used to produce energy, but generally, carbohydrates are the main form or main source of energy production. Glucose is specifically catabolized or broken down by one of two processes: cellular respiration and fermentation. So we've gotten a glimpse of metabolism, and we know that our cells use energy that's in food to make ATP, adenosine triphosphate. ATP powers almost every activity within our bodies, within our cells, as well as the cells of microorganisms that require energy. There's a process called cellular respiration that occurs within the mitochondria. Remember that the mitochondria is an organelle within our cells that is involved in energy production. So cellular respiration occurs in mitochondria, and this is where ATP is produced. This process is called aerobic respiration. So cellular aerobic respiration or aerobic cellular respiration. Two terms I want you to that I want to define for you. Aerobic, Aerobic, simply just means that oxygen is involved. Conversely to that, there is something called anaerobic, so Aerobic. Anaerobic activities do not require oxygen to be present. So aerobic activities or reactions require oxygen to be present. Anaerobic activities do not require oxygen to be present. As previously stated, all cells need ATP. So aerobic cellular respiration is a process by which glucose and oxygen is consumed or taken in, and then it's broken down to produce carbon dioxide, water, and in that process also energy is released. So the breakdown of biomolecules, nutrients, okay, to carbon dioxide and water. You have your glucose and oxygen you consume, and then your end products are going to be carbon dioxide, water, as well as energy in the form of ATP. Now, oxygen is acquired and carbon dioxide is released during this process. What we see here in cellular respiration is the exact opposite of what we may see in photosynthesis, where plants will take carbon dioxide and water to now produce food in the form of glucose, and they will give off as a waste or byproduct oxygen. This process of cellular respiration actually occurs in the mitochondria. I drew a very crude drawing of a dissected cell, if you will, that had, a cross-sectional cut. And I said inside that cell, you have all these other little things, which are organelles, and all these organelles have very specific roles that they play. Well, again, the one organelle called the mitochondria actually plays a role in energy production, and that energy being produced in the form of ATP, adenosine triphosphate. But again, it also has its own interior structure. It has different little folds in it called cristae, but it also has ribosomes. And if you recall, I believe we discussed the fact that ribosomes play an important part in protein synthesis. So if mitochondria have their own ribosomes, that also means that mitochondria can produce its own proteins because in addition to the ribosomes, it also has its own DNA. And remember, one of the functions of DNA is not just to carry out genetic information, but to also provide the recipe and everything that's needed to produce proteins. So mitochondria are capable of producing their own proteins that they need for their various processes because they have their own DNA and they have their own ribosomes. So I kind of briefly mentioned that cellular respiration gives off carbon dioxide as a byproduct, and photosynthesis gives off oxygen as a byproduct because it uses carbon dioxide in its process in order to produce food, that food being glucose. So how are cellular respiration and photosynthesis related to breathing? Well, we inhale oxygen, and that oxygen is required for aerobic cellular respiration. It travels to our mitochondria inside our cells. When that oxygen goes through all of the processes through cellular respiration, when we exhale, the byproduct or waste that is generated at the end of the process of cellular respiration is carbon dioxide. It's released into the atmosphere from the mitochondria. Well, that carbon dioxide now that's in the atmosphere is actually absorbed by plants to be used in photosynthesis, where it will go through its process to produce glucose, which can be food that, is consumed. And once it's finished going through its process of photosynthesis, it will release as a byproduct oxygen back into the atmosphere, and we'll inhale that oxygen, and then it'll go through cellular respiration, and we exhale carbon dioxide. So it's a cycle. It's a cycle. So cellular respiration and photosynthesis are very intricately related in this cycle. I'm not going to spend a lot of time on photosynthesis other than just basic definition and just giving you a picture so that you have it in your mind. Photosynthesis is a process by which green plants and some other organisms, because there are some bacteria as well and algae that are photosynthetic, they use sunlight to synthesize or make food from carbon dioxide and water. Photosynthesis in plants generally involve the green pigment chlorophyll, and we've already stated that oxygen is produced as a byproduct or a waste product of photosynthesis. So whenever we see the word synthesis, it just means to make something or produce something. So here we're going to just talk briefly about ATP synthesis. Also note ATP synthase to the right, that little molecule. And remember, whenever we see ASE on the end of something, we know that it is an enzyme. So it is something that's driving the reaction forward. So potential energy from food molecules is used to make ATP. Remember that energy is stored in the bonds of those molecules. An enzyme attaches, it's going to attach to the active site, a phosphate group to ADP to produce ATP. ADP being adenosine diphosphate, so two phosphates to produce now adenosine triphosphate, 3-phosphate. ATP production requires energy input because something is being synthesized. It's a series of redox reactions. So the synthesis of ATP is a series of redox reactions. So there are electrons being donated, accepted, donated, accepted. It does not happen all at once. It's a series of reactions. Chemical bonds and atoms and glucose are rearranged one step at a time. Tiny bit of energy is released at each step of the process. Energy is stored again in chemical bonds of ATP, and some of this energy is released as heat. So just looking a little more closely at this cellular respiration process that takes place within the mitochondria of our cells, depending on the text that you use, some texts will say that cellular respiration starts with glycolysis. Your text says that cellular respiration starts with the synthesis of acetyl-CoA. But in order for us to get to that process of acetyl-CoA, first that glucose has to be broken down. Glycolysis, whenever we see lysis at the end, it means split or break down. So glycolysis provides the precursor, so whatever comes before that acetyl-CoA is produced. That precursor is pyruvic acid, which is also sometimes referred to as pyruvate. So in looking at cellular respiration, three main steps. So the synthesis of acetyl-CoA, which we get that from pyruvic acid. That pyruvic acid comes from the direct breakdown of glucose. Glycolysis always starts with a process called the Embden-Meyerhof-Parnas pathway, and we're just going to say EMP. So generally, before glycolysis process even starts, or at the very beginning of glycolysis, it's a little prep pathway that it goes through. It's called the EMP pathway. Hold on to that thought because I'm going to make it make sense a little later on. Once we leave that synthesis of acetyl-CoA phase, we go into what's called the Krebs cycle, and then from the Krebs cycle, electron transport chain. So we're going to harvest potential energy from food molecules in order to make ATP, and that's going to happen via this process of cellular respiration. To drive a point home that I already mentioned, cellular respiration relies on oxidation reduction reactions. Release energy by oxidizing glucose and reducing oxygen. Since oxygen strongly attracts electrons, the reaction is downhill, therefore it releases energy. So again, the stages of cellular respiration, I have here glycolysis being the first, but we know that basically glycolysis is pretty much the precursor because it actually produces the acetyl-CoA from pyruvic acid that will enter now into the second stage of cellular respiration, which is the Krebs cycle. Krebs cycle is a derivative of pyruvate is oxidized. Carbon dioxide is released, more energy is transferred to electrons. So during the Krebs cycle, a derivative of pyruvate is oxidized, that being acetyl-CoA. Carbon dioxide is released during the Krebs cycle, and more energy is transferred to electrons. Those electrons now will enter into the electron transport chain. So energy from electrons is used to form ATP. Oxygen is going to be the final electron acceptor in this process. So glycolysis is basically the breakdown of sugar. Glyco standing for sugar, lysis to break or split. That whole process of glycolysis breaks down glucose to pyruvate. Pyruvate is converted into acetyl-CoA, which then enters the Krebs cycle. Now, glycolysis, even though I tell you that the whole cellular respiration process takes place within the mitochondria, technically that's true because it's not until after the pyruvate is formed that we are officially whole hog into cellular respiration. But this precursor stage, which is very closely linked, and that being glycolysis, does not occur inside the mitochondria. It occurs within the cytosol of the cell. So remember, inside the cell, you have the organelles, but you have a liquid portion also. And the cytoplasm is everything, the organelles, the liquid portion. But if we just talk about the liquid portion alone, that is called the cytosol. And when we talk about glycolysis, two main stages are what's called energy investment, so putting energy in, and energy harvest, taking energy out to use it, the utilization of energy. So glycolysis actually splits glucose. It will take one molecule of glucose and then split it into two of these three carbon molecules called pyruvate or pyruvic acid. It'll convert these two molecules of ADP. It will convert two molecules of ADP to ATP. Then electron carrier NAD plus will pick up electrons, forming two molecules of NADH. Don't really expect for you to remember that, but what is relevant is that you understand that glycolysis is just the breakdown of glucose into pyruvic acid or pyruvate, which will later be converted into acetyl-CoA. This schematic is just showing you pyruvate or pyruvic acid moves into the mitochondria. Once it's formed, it moves into the mitochondria, and then it is converted into acetyl-CoA before it enters that next stage, which is the Krebs cycle. So if you'll recall, or just look back at the slide that had the cross section of the mitochondria, and there are different parts of the mitochondria, different components that make it up. Pyruvate moves into the mitochondrial matrix. So that's one of the parts within the mitochondria. The two molecules of pyruvate are converted into two molecules of acetyl-CoA. The electron carrier NAD plus picks up electrons, forming two molecules of NADH, and then two molecules of carbon dioxide are released. So remember, after acetyl-CoA is obtained or produced as a result of glycolysis, breaking down the sugar and forming it into pyruvic acid or pyruvate, and now that pyruvate being converted into acetyl-CoA, acetyl-CoA from the breakdown of pyruvate produced during glycolysis will now enter the Krebs cycle. During the Krebs cycle, an oxidation reaction occurs and there is a release of carbon dioxide. So carbon dioxide is produced and released during the Krebs cycle. Energy is trapped now as ATP, NADH, and FADH or FAD. So again, we say ATP stores energy and it stores energy in these bonds. Well, if it's storing the energy in the bonds, how are we getting it out of storage? Well, we're getting it out of storage by breaking some bonds. So removing a phosphate group via hydrolysis, hydrolysis just means the breakdown by water, it will release that stored energy that's within the bonds. Okay, just a reminder, an exothermic reaction is involved in breaking the bonds. So what happens is when that exothermic reaction occurs to break a bond of ATP via hydrolysis, the ATP now will lose one phosphate and becomes ADP, adenosine diphosphate, so just two phosphates. An endothermic reaction will cause that phosphate bond to reform later on. Now, in this process or hydrolysis and breakdown of ATP, in order to get that stored energy out, you have hydrolysis coming in, breaking one of the bonds of ATP, so one of the phosphates are broken off. And when that one phosphate is broken out, simultaneously there is some energy released as well. So that's how we're getting the energy out of these bonds via hydrolysis of ATP to break off one of the phosphate molecules by breaking the bond and then releasing not only that one phosphate, now forming adenosine diphosphate, ADP, there is also release of energy at that stage. So there's something called an electron transport chain that's within the cell membrane, and the function or job of that electron transport chain is to harness or capture the energy from electrons. So NADH and FADH2 establish what's called a protein gradient, so a concentration of protein ions, and the mitochondria will actually harvest that energy as ATP. In other words, they'll gather it and store it as ATP. So, electron transport chain in aerobic respiration, remember the word aerobic just means that oxygen is involved. Oxygen is the final electron acceptor. It combines with hydrogen ions to form water, H2O. ATP is generated during aerobic respiration, and the intermembrane and intermembrane compartments of mitochondria is where this aerobic respiration takes place. So let's talk about metabolic diversity and what does that mean? Well, we know that metabolism is basically just a sum of all reactions that occurs within organisms and microorganisms. And under that umbrella of metabolism, we have catabolism and anabolism. Well, glycolysis is usually the first step in the catabolism of glucose, the breakdown of glucose. And it usually starts with that EMP pathway. Remember, I talked about that to put a pin in that, remember that. But you have some organisms that have an alternate pathway. So instead of using that EMP pathway as their precursor to enter into glycolysis, they instead use another pathway called Entner Durdorf pathway, or ED, simply ED, may be used instead of an EMP pathway. Some microorganisms will use either or. They'll either use the EMP pathway or they'll use the ED pathway to enter into glycolysis to break down sugars. But you've got some microorganisms, for instance, like E. coli, they can use actually both of those pathways simultaneously. And not many organisms can do that, but there are a few. There's also a totally different pathway from EMP or ED, uh, because again, we're talking about those pathways being precursors to glycolysis. You have a process that's totally different from uh, glycolysis that's called pentose phosphate pathway. And it's a pathway all within itself that can be used to actually break down or catabolize glucose. And just that surface level knowledge is all you need to know about that, just that basic definition or the basic knowledge that there are different metabolic pathways that can be used or that are used to start the whole process of glycolysis, which is the breakdown of glucose. So some of you may have heard the word fermentation, and in everyday use, fermentation is simply the production of alcohol from sugar. But in microbiology, fermentation is the partial oxidation of sugar to release energy. So what do I mean, the partial oxidation? Um, we'll talk about that. Fermentation uses organic molecules within the cell as the electron acceptor. It also oxidizes NADH plus and NAD plus in the reduction process. So just to backtrack a little bit, we've already talked about getting energy from our food, and that energy or those nutrients and molecules now go into this metabolic pathway called aerobic respiration. And different foods, of course, are processed in different areas or digested in different areas of the body, of different organs. So carbohydrates will go through the digestive system and they're converted to glucose. Proteins, the amino acids within proteins can be converted to pyruvate instead of glucose being converted into pyruvate. And then you have fats and lipids, which are digested in the small intestine. These components of fats can also be converted to pyruvate because remember that pyruvate is that precursor that's going to be converted into acetyl-CoA, which will then enter the Krebs cycle, moving that whole process of aerobic respiration forward. So cellular respiration is not always aerobic. Remember, aerobic means that it requires oxygen. It can be anaerobic, which means it doesn't require oxygen to be present. So there are certain organisms that can survive in the absence of oxygen or in the presence of very little oxygen. In these particular cases, glycolysis produces ATP without requiring oxygen. So, in the case of anaerobic cellular respiration, glycolysis can produce energy in the form of ATP without requiring oxygen. So, in anaerobic respiration, in other words, no oxygen is required. There's also no electron transport chain. That's what ETC stands for. Because remember, we have like the first step of synthesis of acetyl-CoA, then we move to the Krebs cycle, then we end up with the electron transport chain where these electrons are being donated and accepted. Well, in anaerobic respiration, when there's no oxygen involved, those steps don't occur. The only step that occurs is glycolysis, and glycolysis, therefore, now in those type of organisms or those situations is the only source of energy. It is the only thing that's producing ATP. So glycolysis is the only thing that produces energy or ATP in fermentation. Fermentation allows glycolysis to produce ATP, but it only produces it in small amounts. There's just two ATP molecules that are produced in glycolysis, whereas in the entire aerobic cellular respiration process, 36 molecules of ATP are produced. Many prokaryotes and some eukaryotic cells use fermentation. So, in other words, they're able to utilize the process of glycolysis to produce energy in the form of ATP, very little, without the presence of oxygen. There's no Krebs cycle or electron transport chain in fermentation. Fermentation allows glycolysis to continue producing small amounts of ATPs because if you think about it, it's better to have a little energy than no energy at all. So many times I know, or not many times, I know some of you all are aware, have a sense of awareness, like with exercising and what have you. And then sometimes what can happen is we can exercise and actually temporarily consume available oxygen in our muscles, or our muscles will consume the oxygen that's available to them at the time. And you don't want your muscles to stop working, they still need energy. So, what will happen is the process of lactic acid fermentation will occur. So, lactic acid will build up in those muscles that have depleted oxygen so that the muscles can continue to work, but Again, they need energy. And where they're going to get the energy from, because we've used up all the oxygen with exercising so hard, they're getting it from fermentation, lactic acid fermentation, because no oxygen is required for fermentation to produce energy in the form of ATP. Now, again, it's just a small amount of energy, smaller than going through the entire cellular respiration process, but it's still energy. Little energy is better than no energy. And remember, only glycolysis is actually at work in the fermentation process. There's no Krebs cycle, there's no electron transport chain, so you don't get the whole gamut of 36 ATPs. You're only going to get a small amount until oxygen is restored to those cells. So we see that happening in muscle cells when we exercise or over exercise. And just in case I did not clearly state it, in the cellular respiration process, the aerobic cellular respiration process, one glucose molecule can produce or yield 36 ATPs, whereas glycolysis by itself will only yield 2 ATPs. Okay. And then those two ATPs now will be used to convert pyruvate into acetyl-CoA, which now goes into the Krebs cycle, producing more or two more ATPs, and then eventually we see a whole set of biochemical processes where electrons will enter into the electron transport chain, and we end up with 34 ATPs, and then there's a couple of electrons lost during that whole electron transport process, and the grand total sum after everything has been done is the production of 36 ATPs in aerobic cellular respiration. But again, in glycolysis, that's used to produce ATP during fermentation, you only have two ATP molecules being produced. Now, we've spent a lot of time on catabolic pathways of breaking down carbohydrates or sugar, glucose, etc. But there are other catabolic pathways that exist, and we're not going to get into them other than just define them. There's what's called beta oxidation, and it's used to break down fatty acids. There's deamination, and basically what deamination does is it kind of strips away amino acids during the process of breaking down proteins. You have the large macromolecule, which is the protein. You have specific enzymes called proteases that will come in and break down the large protein molecule into its individual amino acid parts. And then the deamination process is actually the removing of an amine group from the amino acids to further break them down during the whole process of breaking down protein. So let's revisit our case study. The doctor confirms that the infection has spread to deeper tissues. She notes areas of necrosis. Necrosis just means dead tissue, okay? The infection has not responded to antibiotic treatment. So they've given this young man antibiotics. He hasn't responded. When we state that he hasn't responded, that just basically means the antibiotics aren't working. They're not doing any good. Brad's heart rate is elevated, his blood pressure is dropping, and he's becoming quite confused. So now surgery is performed to remove this dead tissue, this necrotic tissue. After they've surgically removed the necrotic tissue, additional antibiotic treatment is given to this young man post surgery, and it is given through an IV, so intravenously. Once all that's done, now Brad makes a full recovery. So if you remember at the beginning, we took blood cultures and sent those to the lab to be tested. But additionally, this dead tissue that was surgically excised was also sent to the lab to be tested. At the end result, it was determined that he had an infection caused by a bacteria called Streptococcus pyogenes, which is a group A streptococcus. We'll talk more about those a little bit later. We know that since it's streptococcus, we know something about its morphology because we've talked a little bit about shapes of microorganisms. So that means it's in chains and it's circular or spherical. It is gram positive. That means it has a thick cell wall. And it can also cause conditions like strep throat. But in this particular case, it caused what's called necrotizing fasciitis or flesh eating disease. So, in summary, various metabolic pathways are involved in utilization of nutrients and various molecules to sustain our life. But also, many of these same metabolic pathways are used by pathogenic organisms. So, in other words, organisms that can cause infections and disease to produce energy to carry out the ability to infect us. Some of these metabolic processes are aerobic, which means they require oxygen. Some of them are anaerobic, they do not require oxygen, as we saw in fermentation. Enzymes drive the biochemical reactions of these metabolic processes forward, and we remember that whole enzyme substrate specificity when we talk about enzymes. Most enzymes are proteins, but there are some that are made of RNA called ribozymes. So, again, go back, review the slides, and study the material. I think it's pretty interesting material, and I hope you enjoyed it.